distinși membri ai Academiei de Științe a Moldovei și membri desemnați ai secțiilor de știință Academiei de Științe a Moldovei. Excelență, domnule ambasador al Statelor Unite ale Americii, stimați tineri, doctoranți, studenți, liceeni, onorată asistență, astăzi este o zi deosebită pentru Academia de Științe a Moldovei și pentru comunitatea științifică a țării noastre. Ne onorează cu prezența o mare personalitate a timpurilor noastre, profesorul Randy Schekman de la Universitatea California, Berkeley, din Statele Unite ale Americii. Laureat al Premiului Nobel pentru medicină, savant cu rădăcini basarabeni. Părinții mamei provin din orășelul Lipcani, unde îi sunt înmormântați bunicii, prin urmare străbunicii profesorului Schekman, ceea ce l-a determinat să declare că este datoria lui sfântă să viziteze Republica Moldova. Vizita pe care o actualmente o întreprinde profesorul Schekman la invitația Academiei de Științe a Moldovei reprezintă, după propria mărturisire, prima întoarcere la baștină a unui membru al familiei în decurs de circa 100 de ani. Distins auditoriu, în ultimii ani, Academia de Științe a avut o colaborare fructuoasă cu profesorul Randy Schekman. La 28 aprilie 2020, domnul profesor a deschis simpozionul științific Pandemia COVID-19, suport științific medical sau ce trebuie să cunoaștem despre coronavirusul SARS-CoV-2 și pandemie, convocat de Academie de Științe a Moldovei în colaborare cu Universitatea de Stat de Medicină și Farmacie Nicolae Testărițanu. A susținut online, de la tribuna Academiei de Științe a Moldovei, o serie de prelegeri publice despre importanța științei fundamentale în medicină. În ultimii 4 ani, profesorul Schekman a acordat interviuri și a participat online la emisiuni televizate, în cadrul cărora a adus informații de ultimă oră cu privire la pandemie și problemele ce țin de răspândirea coronavirusului SARS-CoV-2 și elaborarea vaccinurilor. Luând în considerare această colaborare fructuoasă cu cercetătorii din Republica Moldova și deschiderea sufletească a domnului profesor Randy Schekman spre noi, adunarea generală a membrilor titulari și membrilor corespondenți ai ASHM din 28 mai 2021 l-a ales unanim în calitate de membru de onoare al Academiei Științe a Moldovei. Astăzi, înainte de a prezenta lecția publică, Calea mea spre Premiul Nobel, îl invităm pe domnul profesor Randy Schekman pentru a-i înmâna însemnele de membru de onoare al Academiei de Științe a Moldovei. That's the document, so scholarly member of the Academy of Sciences of Moldova is compared to Professor Randy Schekman from the University of California, Berkeley, for his unestimated Sciences of Moldova and researchers from the Republic of Moldova. So please, pass on to the lecture. I would like to give the floor to His Excellency, Ambassador of the United States to Moldova, to congratulate our please.
Thank you. Bună ziua. Nice to see everyone. It's wonderful to be here at the Academy of Sciences of Moldova on such a special occasion. President Tiginian, esteemed members, thank you for asking me to participate in marking what I would say is a historic lecture. Not only do we see the award of joining as an honorary member of the Academy of Sciences, but actually someone said you don't often have the honor to meet a Nobel laureate. I've never met a Nobel laureate. This is my first time, so I'm very, very grateful to be here. I could not be prouder of the fact that this particular Nobel laureate is an American citizen who has strong and deep roots here in the Republic of Moldova. So that's another exciting part of this day. What's happening here today is a great example of people-to-people connections and ties, the most important part of diplomacy. And as an ambassador, I spend a lot of time in the world of policy, security, trade, so many other areas. But none of that is possible, or it doesn't even matter as much unless we're talking about people to people and our citizens who are building relationships with each other, one relationship at a time. So thank you, Dr. Schekman, for all you are doing to build these ties that bind the people of the United States and the Republic of Moldova together, as partners, and also as people who cherish living in vibrant democracies. I know most of the audience here, you're all familiar with Professor Schekman's unparalleled achievements in the worlds of science and research and in academia, and of course, medicine at some of America's finest universities. His extensive contributions were worthily celebrated in one of mankind's highest honors, the Nobel Prize in Medicine or Physiology, and that was awarded in Stockholm 10 years ago. So Dr. Schekman, it gives me great pleasure to welcome you here officially to an institution and to a country which we both know is incredibly special. Thank you very much.
Ladies and gentlemen, before we pass to the presentation of the public lecture, my career path from Scum to Stockholm, according to our tradition, we will listen to the musical composition Academia, Symbol of the Nation. Academia, Simbol al Națiunii. Compoziția muzicală, autor, profesorul Dear Professor Schekman, now you have the floor.
Well, thank you very much, Professor Tiganyanu. Let's see, okay. After this very moving introduction, I can only imagine what my grandparents would have thought if they'd lived to see this day. As you may have heard, my grandparents were born in the village of Lipkan in the early part of the 20th century. They emigrated from Lipkan, where my grandfather was a tailor in the Romanian army. They immigrated to the U.S. in 1927, and my mother was born in Minneapolis, Minnesota. My grandparents nor my parents ever came back to visit the village or to visit Moldova, so I'm the first person to return to this country in nearly 100 years. So it's going to be a very emotional occasion for me this week at Professor Tiganyanu's introduction. So let me tell you briefly about my journey. I've given a slightly different title to this lecture than Professor Tiganyanu mentioned. I grew up in Southern California in an area that was very suburban at the time, and I developed an interest in microorganisms when I was a very young child. I had a toy microscope that I got when I was maybe 11 or 12 years old that I used to examine a drop of pond scum from a nearby creek, and I was able to see the amazing life that one can see even with the small magnification of a primitive microscope. One evening at dinner, I told my parents about my exploration of these microorganisms, and my father was rather skeptical that I could see anything in this toy. So I resolved that evening to save my earnings to buy a student professional microscope. And after some effort at mowing neighbors' lawns and babysitting and delivering newspapers, I purchased, as you see on this screen now, a Bosch & Lomb microscope that was my pride and joy throughout my years remaining at home in high school. Every year I mounted a science fair project having to deal with microorganisms, and brought it to the county fair and to the state fair, and developed my passion for science, particularly the science of microorganisms, which I took to me to college and then to graduate school. Of course, the microscope got left behind at home, but I'll say more about that later. In graduate school at Stanford University, I worked with a very famous biochemist by the name of Arthur Kornberg, who'd won his Nobel Prize for discovering an enzyme that copies DNA to help make chromosomes. But most importantly, I learned from him how to take a complicated biological problem and to tease it apart into its mechanism by isolating the molecules that execute a process. After graduate school, I did postdoctoral work in Southern California, and I was introduced to the work of the great Romanian cell biologist George Palade, who did his pioneering work in New York at the Rockefeller University, where he was able to discover the process that cells in the pancreas use to capture molecules for export from cells, molecules like insulin. So let me take you through on a journey through the cell, in part through Dr. Palade's work, to tell you about the problem that I investigated in my own laboratory. So just a bit of a primer about the molecules that are in a biological membrane. Here you see, in several cartoons and a microscope image, the double-track appearance of a biological membrane that surrounds a cell. And as you'll see later on, cells in our body are much more complicated than this. They have many membranes, but for simplicity, let's just consider the nature of the membrane that surrounds a cell. It consists of two layers of molecules called phospholipids. These are molecules that have hydrophilic surfaces that face the outside of the cell and the inside of the cell, and then they have very hydrophobic fatty acid side chains attached to the head groups that make a very apolar hydrophobic interior to the bilayer. By itself, these molecules would insulate a cell and prevent any small molecules' passage one way or the other. But membranes have evolved to capture protein molecules that impart personality in the form of different channels or receptors embedded in the membrane. So these green blobs here may suggest, for instance, a channel through which small molecules may move specifically, or receptor molecules to which hormones may bind to elicit some physiologic process within cells. So membranes are very complicated, and each membrane in a cell has a different personality. This was realized by the pioneering work of George Palade, shown here in the later part of his career, where he became a professor at the University of California, San Diego. He's seated behind an instrument that he made a very powerful tool to inspect the membranes in cells. This instrument, called the electron microscope, was first created by German engineers to inspect physical materials, but it became possible through the efforts of Palade's laboratory in New York to inspect thin slices through cells that had been preserved chemically so that they could resist the flow of electrons generated down this chamber that bombard thin slices of tissue and penetrate through the tissue where the tissue has no particular heavy stain taken by particles. The electrons are diffracted, or they bounce off of densely stained particles, and so you can visualize in a thin slice through a cell dark and light areas that represent the cytoplasm versus membranes and particles, dense particles, and I'll show you examples of those. Anyway, over many years, Palade perfected the techniques that allowed us to visualize the cell and to understand its ultrastructure. Here's an example. Let's see if I can move this. Yeah. Here's an example of a cell that was understood in great detail by Dr. Palade. Very important cell in our pancreas. It's called the beta cell. It's the cell that organizes the production and secretion of insulin. I'm hearing a recording of myself. This is a thin slice of a pancreatic beta cell. You see the nucleus of the cell where the chromosomes are housed. You see various membranes spread around the cytoplasm, but most importantly, you see granules shown here that have a dense crystalline arrangement of insulin molecules that have become enveloped by a biological membrane that surrounds each granule. And these are housed in the pancreatic beta cell. They build up in between meals, and when you have a meal and you start to digest polysaccharides, glucose molecules are produced, released into the bloodstream, and that elicits a process of secretion by where something's not working now. The cursor is not following my instructions. Somebody know how to operate this instrument here? The cursor has its own mind. First of all, I want to get rid of that thing. Oh, there it is. That's the cursor. Okay, get rid of that little screen. Okay. All right. Very good. Thank you. Okay, so when you have a meal and you digest polysaccharides, glucose is released. It floods into the bloodstream, and it bathes the pancreas, and it stimulates these little granules to move to the perimeter of the cell, where by a process of membrane fusion, now it won't move forward. I need help again. pressing all the buttons. There. Has its own mind. So this is a higher magnification view of the slide you saw previously, where a granule, that is a membrane surrounding a condensed granule of insulin, moves up to touch the membrane that surrounds the cell, and at a key moment, stimulated by another hormone, the membrane that surrounds the granule merges with the membrane that surrounds the cell by a process called membrane fusion that results in the ejection of this granule of insulin that dissolves and allows insulin molecules to spread around the body, and that stimulates the uptake of glucose that was produced by digestion into cells of the muscle and into fat cells. So we were able to know this at the level of the electron microscope, but we really didn't know anything about the proteins that are required for the fusion process. Another point where this process of membrane fusion is terribly important is in the brain, or at the junction between nerve cells and muscle cells, where a nerve terminal, shown here, has a number of small membranes called vesicles. These vesicles do not carry insulin. They carry chemical neurotransmitters, such as dopamine or serotonin, or the major neurotransmitter in the brain called acetylcholine. And these vesicles dock on the surface of the nerve terminal, and at a key moment, again, highly regulated, there's a process of membrane fusion where the neurotransmitter is discharged into the gap between, in this case, a nerve cell and a muscle cell, the neuromuscular junction. And these chemical neurotransmitters stimulate the activity of the muscle cell, eventually causing it to contract. Within the brain, there are estimated to be 12 billion different nerve cells, each of which can make up to 1 million different connections. So there are more than 1 trillion different possible contacts between nerve cells, and all of them are controlling this flow of neurotransmitters by a process absolutely dependent on membrane fusion. That communication of chemicals within the brain is what constitutes our thought processes, our emotions, controls our movement. All of that is absolutely keyed into the process of membrane fusion. Well, Dr. Pallotti realized this in his brilliant work, and in 1974 he was awarded the Nobel Prize. Just after the ceremony in Stockholm, he traveled to San Diego, where I had just begun as a postdoctoral fellow at University of California, San Diego, where he delivered a lecture on his brilliant work deducing the stages within a cell that a molecule like insulin must flow, and he wrote this up in a very powerful review article published in Science magazine in 1975. Here's a summary of the experimental results that Dr. Pallotti achieved with his laboratory. He was able to show that proteins like insulin begin their journey by being synthesized by little particles in the cytoplasm called ribosomes. These are like little sewing machines that stitch amino acids one next to another to make polypeptides. Pallotti was actually the first scientist to visualize ribosomes in the cytoplasm of a cell. He showed that molecules like insulin are made by ribosomes on the surface of a membrane called the endoplasmic reticulum. They are threaded across that membrane, and where they build up within the interior so-called luminal space of the ER. The molecules fold. They are then passed step by step through a series of other organelles, eventually to be discharged in granules at the cell surface. Well, Pallotti described the experiments that led to his deduction of this pathway, but what was missing in 1974 was an understanding of any of the molecules engaged in this process. Not a single protein or lipid molecule had been ascribed a role in any of these stages of this pathway. And so I began my career at the University of California, Berkeley in 1976 with the intention of trying to discover the mechanics of this process, to understand which molecules actually were involved at each of these stages, ultimately the molecules required for membrane fusion. What were these molecules and how did that process work? I began by doing something that had not been done in cell biology, at least for this process, and that was to use a simple microorganism, baker's yeast, to try to understand the mechanics of this process. Yeast cells evolved probably about 2 billion years ago on Earth, and the machinery required in our bodies to organize the secretion of insulin evolved 2 billion years ago in yeast, where these single-celled eukaryotic microorganisms figured out a way to organize this process. Yeast cells grow by a slightly different mechanism than human cells, and here's another EM view of what a yeast cell looks like. The yeast cell grows by a budding process. If it's growing in a rich broth, at the beginning of a cell division cycle, a small bud appears on the surface of a mother cell, and that bud enlarges during the next 90 minutes or so, growing step by step until it approaches the size of the mother cell, at which point it divides in half. And so you can grow large cultures, billions of yeast cells, identical yeast cells in a simple laboratory experiment. The virtue of this is that you can use a simple form of genetics to discover the genes that organize any fundamental essential process, techniques that then were not available to study the process in human cells or in other animal cells. They were readily available in yeast. So we began to look at yeast cells to see how they may organize the manufacture and export of their own molecules. Of course, yeast cells don't make chemical neurotransmitters, they don't make insulin, but they make other proteins that allow them to grow and divide and harvest nutrients on the surface of a grape, for instance. So here's an image, a thin slice through a yeast cell. Looks very different than a pancreatic beta cell, but it has all the machinery, especially all those ribosomes. These dots in the cytoplasm here are all ribosomes busy making different protein molecules. There are membranes in the cell. This is a structure similar to the mammalian lysosome. It's called the vacuole. There are membranes that are known to be endoplasmic reticulum membranes, and in other sections you can see the yeast cell nucleus. But what our attention was drawn to in 1976 was a population of small membrane vesicles, similar in appearance to those at a nerve terminal, that are precisely in the region of the cell that is growing and dividing during most of the cell division cycle. We imagined that these vesicles, small membranes, would carry molecules not only to be exported outside of the cell, but they would carry lipids and membrane proteins that would be responsible for building the plasma membrane. By a process of membrane fusion, new pieces of membrane would be inserted that would allow the bud to get bigger and bigger during the 90 minutes that are required to make new yeast cells. Another view that emphasized our hypothesis is shown here. This is a picture of a yeast cell that has been grazed by a fracture plane that cuts right through the surface membrane and then shadowed with a metal replica on top of that section plane. And you can see this bud as emerging from the surface, the rest of the surface, and in the bud you can see these little dimples, little craters, that are the very moment where a vesicle has just joined by the process of membrane fusion. Clearly enriched on the part of the membrane that's growing, getting bigger during the yeast cell division cycle. Well, we had this idea, and then a brilliant graduate student shown here, Peter Novik, one of my first graduate students. Peter and I devised a simple procedure to look for mutations that cripple this process. If you take a culture of yeast cells and you expose it to a chemical that causes mutations, and you plate the cells out on a petri plate, you can grow up colonies that have any of a number of different defects, and you can survey each colony to look for a mutation that causes a block in the export of enzymes that yeast cells need outside of the cell in order to grow, for instance, on the surface of a grape. So we isolated the first such mutation in late 1978, and Peter was able to show that the cells, as they are dying, cause the enzymes that should have been secreted outside of the cell to build up inside of the cell. Quite unusual. We hadn't seen that before. And just by coincidence, Dr. Pallotti visited Berkeley to give 2 honorific lectures in May of 1978. And Peter and the other graduate students organized a dinner in his honor. Peter told Dr. Pallotti about his results, and Pallotti said, well, now you must examine these cells by cutting a thin section to see what's going on in the cytoplasm of the cell. And one of the most exciting moments of my career came shortly thereafter when Peter called me in my office to come down to the basement of the building to look at the screen that he could see in the electron microscope. And here's one such image. You recall a moment ago I showed you a yeast cell with just a small number of these little vesicles in the bud. Instead, in this mutant, as the cell is growing and dying as a result of this mutation, the cell builds up thousands of these little vesicles that can no longer find their way into the bud to fuse. Instead, they spill out and fill the entire cytoplasmic content. And then eventually these cells die. If the cells are allowed to resume growth by changing the conditions that are more permissive, then the vesicles that had accumulated find their way back and are able to discharge their content. Well, this was a very exciting result to us, and over the next year and a half, we were able to isolate hundreds more such mutations, defining a number of genes, not just a single gene, but almost 24 different genes. And years later, we now know that there are more than 100 such genes in the yeast genome. We characterized many other mutants by procedures that I don't have time to describe, and we're able to map out a path that looked very much like what Pallotti had established for pancreatic cells, except in our case, we could show that at each station along the way, there were a number of different genes that have these genetic designations. The first such gene that we found, we called SEC1, but that was only one of many other genes that we were able to show operate at unique positions along this pathway. It became possible to obtain, to clone these genes, to discover the nature of each gene, and to deduce the nucleotide sequence of each gene. And we discovered that SEC1, as well as all the other genes, have counterparts in the human genome. SEC1 is operating right now in this room in all of your brains to organize the process of fusion of vesicles that are carrying neurotransmitters to the presynaptic membrane or at the neuromuscular junction. It is an essential gene in almost all the cells in our body, and it is almost the same gene that we discovered in yeast now over 40 years ago. Now, it was possible because of the conservation of this to understand the unity of biology, and I use the following cartoon to indicate this. On the left is a drawing, a painting of Charles Darwin, and on the right is a cluster of yeast cells, such as you might find growing on the surface of a grape. And I will submit to you that the genes that organize the process of secretion of chemical neurotransmitters is the same in a yeast cell as it was in the brain of Charles Darwin. And that unity of biology is true for many other processes that happen within our cells that have been conserved fundamentally through this essential process of evolution. Well, the fact that yeast cells have the same pathway and the same genes and the same mechanics for protein export as we do suggested to the biotechnology industry, which was growing up in the San Francisco Bay Area, that yeast could be used as a platform for the manufacture of clinically important proteins such as insulin. In fact, I helped a local biotechnology company near Berkeley to harness the gene for human insulin and to introduce that into a yeast cell so that the yeast cell would recognize it as one of its own. And now, many years later, the European country Novo Nordisk manufactures about 1/3 of the world's supply of human recombinant insulin made by secretion in yeast, harnessed in these large fermentation vats in their factories, growing metric tons of human recombinant insulin to replace the sources of insulin that were used to treat diabetics years before the advent of recombinant DNA. Pig insulin was the source then. So this technology started with a purely curiosity-driven interest in the fundamental processes that occur in cells, but whenever one makes that kind of a fundamental discovery, there will be a practical application, an unanticipated practical application. In that case, it was the use of yeast cells to manufacture clinically important drugs. Now, our goal all along was not to do anything practical, but really to understand at a fundamental level how this process works. Having the genes and even knowing the nucleotide sequence of the genes was not enough to understand how this process works at a molecular level. And so, given my training in Kornberg's lab, I decided it would be essential to use biochemistry to understand the nature of the molecules that organize, for instance, the production of these little vesicle carriers. And so I don't have time to tell you about this, but let me summarize some of our findings. It was possible to break open yeast cells and to fractionate the protein molecules necessary to make vesicles using a biochemical approach. And when we did this, we discovered a set of molecules, protein molecules, that are seen to surround and sculpt vesicles that in this case are pinching from the endoplasmic reticulum to make a carrier vesicle. These appeared to be fuzzy coated molecules. Here is a similar preparation. Thousands of uniform vesicles with a electron-dense but fuzzy coat. Subsequent work, however, showed that these seemingly fuzzy coats have a very regular structure. Other scientists were able to obtain crystal structures of the molecules that form this coat that's responsible for pinching vesicles. The coat, which we call COP2, consists of two layers, a set of proteins that bind on the cytoplasmic surface of the ER membrane and recognize protein molecules that are destined to be captured for secretion, and another layer of proteins that form a kind of a scaffold that forms on the surface of a membrane and changes its shape so as to pinch a vesicle to create this coated vesicle. So we knew a great deal about we know now a great deal at the atomic level about this coat, but what happened quite unexpectedly after our discovery was I had a call from a clinical geneticist at Johns Hopkins University who'd been studying with a colleague from Riyadh, Saudi Arabia, a Bedouin family that had a most unusual craniofacial disorder, shown here. These children, who have two bad copies of the relevant gene, are born and mature even though the soft spot on the top of their head never fully closes. They develop cataracts at a young age. They have brittle bones, but they still grow into at least a young adulthood with this unusual malady. What the surprise was when we discovered that that the mutation that causes this pathology is in an invariant amino acid in one of the proteins that we discovered as part of this coat in yeast. Here is a part of the gene of the protein sequence for the yeast protein called Sec23, and two copies of the human gene called Sec23A and B. This particular amino acid, phenylalanine, with the designation F, is absolutely unchanged in this position in every gene copy of Sec23 known in the database. But it is changed in these patients to a leucine residue instead of a phenylalanine residue in a part of this molecule that rests on the cytoplasmic face of the ER membrane, touching the ER membrane, and this residue seems to be important, and when mutated, it causes the coat to be defective. Defective so much so that when you look at cells from one of these kids and you compare it to normal skin cells, or even to cells of one of the parents who have one bad copy of the gene and one normal copy and who are disease-free, you see this really striking difference in appearance. Here in the top panel is a thin slice through a normal human skin cell showing the ER membrane with ribosomes and some granular material in the inside. The middle panel is of one of the parents, a carrier, with some distorted ER membrane. But look at the section through the skin cells from one of the children. The ER membrane is enormously distorted and bloated. And we know now that it has the difficulty secreting a molecule called collagen, which is a major structure-forming protein on the surfaces of our skin cells. And these skin cells, harvested from one of the kids, show this quite striking defect, purely as a result of a change in that one amino acid. Well, this, of course, was a very interesting discovery, and it continues along a line of such discoveries that we've been able to make by using a combination of genetics and biochemistry, and I would have been happy to live out the rest of my career uninterrupted by other things in my life, but that was changed in October, now nearly 10 years ago, when I received a call early in the morning from a representative of the Karolinska Nobel Committee awarding me the Nobel Prize. And so my family were quite happy, and my 86-year-old father was, I was able to call him early that morning to share the good news. We all traveled to Stockholm, and the main event in Stockholm is a celebration in the concert hall where Nobel laureates seated on the left, the front row being the newest Nobel laureates and the back rows being previous laureates from previous years, and on the right, the royal family, together with members of the various Nobel juries, sit for the celebration. I was able then to receive the Nobel Prize from the King and the medal shown here. But perhaps the biggest surprise came when, shortly after the announcement of the Nobel Prize that year, I received, along with all the other laureates, a request to donate some part of our, some piece of equipment that may reflect the discovery that was the basis of our Nobel Prize. I didn't think sending them a Petri plate with the colonies of yeast would be at all interesting, but what I thought instead was to send them that old microscope that I used to begin my studies when I was in high school. My parents saved that microscope, and they sent it up to me and my family in Northern California. Neither of my children ever showed any interest in science, so the microscope sat and collected dust in the closet. I dusted it off, sent it to Stockholm, and now you can visit my microscope, shown here, together with a little caption in English and in Swedish about how I was able to acquire this when I was very young. So that's the story of my journey, and I'm happy to have been able to share that with you this afternoon. Thank you.
Dear Professor Schekman, thank you very much for this wonderful lecture. And the lecture is open for discussion. In case of questions, we have many young people and researchers here who would like to put a question. Dear Professor, I remember you wrote about impact factors of journals. You have had your own opinion about this. And actually, the question is the following. Do you think that it's true in case one succeeds to publish a paper in a journal with high impact factor, it automatically means that the paper is of very high quality?
So thank you, Ann. So this is a conversation that requires a longer discussion, but let me just briefly introduce the topic to you. Some of you will be aware that what we do in this business is we publish our work. That is the currency of our intellectual exchange. And over the years, the process of publishing scientific work has changed quite dramatically. It used to be a process organized entirely by academics for the benefit of scientific societies. But it's become a very large commercial enterprise as well. The largest publisher of the scientific literature are now two large corporations, Elsevier and the Nature Publishing Group. And the process become so complex that some people felt it would be necessary to have a measure, some quantitative measure of the influence of a piece of scholarship. Nearly 50 years ago, such a measure was created by an organization in the US called the Institute for Scientific Information. It was a simple calculation called the impact factor, journal impact factor, where the number of citations to papers in a particular journal computed over the period of 2 years divided by the number of papers published by that journal would be somehow a measure of the influence of that journal. The higher the number, the more the influence or the more the impact. This number was created in the first instance to be used by librarians to decide which journals to subscribe to. It's a reasonable thing where a librarian would need to know how popular a journal was to make it available to a library. But unfortunately, over the years, it has evolved into a measure of scholarship. Now, journals sell themselves on the basis of this impact factor. And if you know how the number is calculated, if you know really what it means, you would appreciate that it has nothing to do with a measure of scholarship. Many very important papers don't get cited at all in the first 2 years after they've been published. A truly original result sometimes takes years before the scientific community embraces, adopts, and then attempts to replicate the work. So measuring it over a narrow window is a distortion. It only measures the impact of papers that are very popular at the time that they're published, not papers that are really of long-lasting redeeming value. So I have argued as much as possible against the use of this number. It is not a measure of scholarship. It's a measure of commerce more than of scholarship. But unfortunately, institutions and university administrators and academic review bodies often look at a scholar's output in terms of the impact factor of the journals in which the work is published. This is an absolute distortion, and it should not be used in any institution to evaluate anything other than whether the journal belongs in the library.
Thank you very much. I will say a few words in Romanian. Deci, domnul profesor Schekman a explicat că impact factorul ăsta, deci, așa, deja pe tradiție se ia ca o măsură a calității, dar de fapt aici nu este totul chiar așa cum scriu revistele, fiindcă foarte multe articole în care se descriu fenomene noi, sunt descoperiri, deoarece nu sunt alte echipe care se ocupă cu așa probleme. Dacă e ceva nou, înseamnă că e nou. Și alții, și nu sunt citeri, ani întregi nu sunt citeri. Deci asta este o problemă. Iar cei care se ocupă cu lucruri care de acum, cunoscute la toată lumea, ei o să aibă citeri. Toți, foarte multe echipe se ocupă cu asta. Deci dumnealui a fost unul dintre primii care a abordat această problemă. Fiți atenți! Asta este o capcană pentru a diminua importanța unor lucrări absolut noi. Eu singur am văzut și am întâlnit cercetători care au fost și cazuri când a prezis ceva absolut nou și de 35 de ani lucrarea n-a fost observată. Pe urmă, când a fost observată, a fost, desigur, delta funcția în sus. Deci este o problemă la scară internațională și domnul profesor Schekman e unul din primii care a abordat această problemă și foarte serios. Și a deschis o revistă nouă în care a permis, în primul rând, celor tineri să publice e-life, electronic journal e-life. Sunt întrebări? Domnul academician Gulea, vă rugăm la microfon. Dar să vă vadă lumea, veniți aici. Veniți aici să vă vadă, uitați-vă, noi avem foarte mulți în spațiul online.
In your lecture, you talk about George Palade. Don't you have any contacts, scientific contacts with George Palade or him collaborations during your life?
Yes, of course. I'm happy to tell you about as I said, I didn't study directly with him, but he was a colleague of my Professor Kornberg. They were friends, Kornberg had done a little sabbatical study in Pilates' lab the year before I started graduate school. I met Pilates when he visited Stanford while I was a graduate student. And then I met him directly when he came to Berkeley just after we isolated the first secretion mutant. And I had the opportunity to tell him about our work. He was at that point surprised that yeast cells secrete proteins. He wasn't aware of this. So I was pleased to be able to tell him that. And more importantly, my student, Peter Novick, had dinner with him that night and told him about his work. And that was when Pilates suggested that we use the electron microscope to look at the mutants, and that generated that wonderful image that I showed you of a cell filled with vesicles. Subsequent to that, I had the pleasure of interacting with Dr. Pilates professionally on symposia, and on 2 occasions near the end of his career. And then, because of my relation with him, I was asked to write the obituary in Science magazine when he died. So I feel he was a patron saint of my field, and I value him as a good friend and colleague. Really a quite remarkable man. Probably no one in this room has had the opportunity to meet him, but he was a scholar of deep distinction in many different disciplines, including Roman military history as well as cell biology.
Thank you. So, are there other questions? I do not see it. Academician Mihai Cimpoi wants to say a few words. Dear Professor, maybe you take your place, because I will translate for you. Da. Domnul profesor, sala cunosc limba română.
Stimate domnule profesor, în primul rând vreau să vă adresez un sentiment de recunoștință pentru vizita dumneavoastră, pentru prestanța dumneavoastră științifică și pentru faptul că nu uitați rădăcinile, nu uitați tradiția, nu uitați cei și poeții noștri. Eu stau chiar acolo cu niște artiști, cu niște poeți, numesc izvoare, atât de frumos, rădăcini și izvoare. Eu sunt, domnule profesor, originar din Lipcani, de lângă Lipcani. Lipcani este un adevărat centru de cultură evreiască. La Lipcani s-a născut clasicul literaturii evreiești de limbă jidiș, Eliezer Steinberg. La Lipcani au activat foarte mulți scriitori evrei care s-au înscris organic și în cultura românească. Un grup de scriitori de Lipcani, de evrei născuți la Lipcani, au format în Statele Unite o societate literară care s-a numit Olimpul din Lipcani. Despre toți acești scriitori evrei și despre scriitorii evrei pe care i-am avut și aici în Basarabia, eu vorbesc într-o carte a mea care se cheamă O istorie deschisă a literaturii române din Basarabia. Deci la pagina 218 o să găsiți absolut amănunte și informații despre scriitorii evrei care au o importanță deosebită și pentru cultura românească. Vă mulțumesc încă o dată.
Okay, so are there any other questions? No questions. I would like to draw your attention that the lecture has been live streamed by the Institute of Development of Informational Society and also by PDSKU. And the links will be available in the future on the portal of the Academy of Sciences of Moldova. That means those who would like to watch this lecture, or those who failed to follow the lecture today, they have the possibility to enjoy the lecture afterwards. Let me thank very much Professor Randy Schekman for his visit to the Academy of Sciences of Moldova, to the Republic of Moldova, and for his very interesting lecture, and actually for many lectures that he presented here and will present in the future. In a couple of days, he will will deliver a lecture at the International Conference on Nanotechnology and Biomedical Engineering. This conference will start on September 20th at the Institute of Labor. And who is interested may, of course, come to this institute to attend his lecture, pure scientific lecture, and there will be, of course, many lectures presented by researchers from various countries, from United States, from Japan, from Germany, and from many, many other countries. Înainte de a finaliza, eu vreau să vă propun, deoarece suntem mulți în sală, să vă propun să facem o fotografie în fața blocului. Acolo sunt mai multe trepte și elevii, studenții să fie în față, cei mai în vârstă sus pe scări, deci și să facem o fotografie pentru istorie cu domnul profesor Schekman împreună. Deci eu vă rog toți în fața blocului, încercăm organizat chiar acum. Vă mulțumesc la toți!